Next session will be HCC 13, Unraveling the Progression of Genomic Anomalies in Hepatocellular Carcinoma, chaired by Dr. Tsutomu Masaki, Japan, Dr. Yoshiyuki Ueno, Japan, Dr. Jian Zhou, China.
Okay, let's start this session. And I'm Yoshi Ueno. We are chairing this session along with Professor Masaki and Professor So. And the first speaker is Professor Nori Kawata. He's very famous.
Thank you.
From Osaka Metropolitan University. And he's giving a lecture about the unraveling the progression of genomic abnormality in hepatocellular carcinoma. Professor Kawata, please.
Thank you. Good morning, everybody. And Mr. Chairman and ladies and gentlemen, it's my great pleasure to give an introductory talk on unraveling the progression of genomic anomalies in hepatocellular carcinoma. I have nothing to disclose. And this is an agenda for my talk today. First, I will give a talk on epidemiology and general aspect of hepatocellular carcinoma, HCC. So this shows the global burden of primary liver cancer in 2020. About 900,000 people were diagnosed with liver cancer and 830,000 people died. Liver cancer is among the top 3 causes of cancer deaths in 46 countries. And the biggest problem is that if current situation persists, it is expected to increase by more than 50% by 2040. So here you see the incidence of hepatocellular carcinoma according to the geographic area and etiology. Mongolia it has the highest incidence of hepatocellular carcinoma, HCC, in the world, and East Asia, as you see here, West Africa and Latin America, hepatitis B derived HCC is the most common cause, and in Japan and the United States, hepatitis C, and in Europe, alcohol. So liver cancer related to metabolic syndrome has been increasing in recent years. So end-stage liver disease like cirrhosis is derived basically from the fibrotic liver, although there is evidence that MAFD and idiopathic cases are slightly less likely to have liver cirrhosis. Therefore, it is very important to understand the molecular events underlying liver fibrosis. So in cirrhosis, a series of genomic and epigenetic events, as you can see here, culminate in the formation of this plastic nodule. And additional molecular alterations provide this plastic cell with proliferative, invasive, and survival advantages and complete the transition to full breakdown of hepatocellular carcinoma, which accumulates somatic DNA alterations. So that promoter mutation occur in about 60% of hepatocellular carcinoma, and other mutated gene affects the cell cycle are TP53 mutation, catenin B1 mutation, ARID1A, AXIN1, AXIN, or CCND1 mutations, as you see here. So then, I'm going to talk about why does hepatocellular carcinoma develop from fibrotic liver. So after a liver injury takes place, there is necrosis and regeneration of the hepatocyte takes place. Continuous cycle of this necrosis and proliferation process foster a chronic liver injury leading to liver cirrhosis. Subsequently, hyperplastic nodules are observed, then this plastic nodule, and finally, hepatocellular carcinoma is occurred in such a cirrhotic liver. Telomerase shortening and loss and/or mutation of p53 and oxidative stress induced genomic instability characterize hepatocarcinogenesis. So fibrotic liver can be said as storeroom of growth factors. So as you see here, the activated stress cell, activated myofibroblast, and inflammatory cells, damaged hepatocyte, cholangiocyte, and L6 are a good source of the growth factors like EGF, FGF, HGF, PDGF, PEGF, and so on. And these growth factors bind to the receptors that are expressed in a hepatocyte, and these growth factors stimulate intracellular signaling pathways like MEK-ELK pathways. So this signaling induced hepatocyte proliferation. In addition, the generated superoxide and peroxynitrite initiate the DNA damage as a logical function. So DNA in the hepatocyte is damaged, mutated. So such kind of liver cancer can be propagated by using the cancer microenvironment, such as by using angiogenesis and inflammatory reaction and fibrotic reactions. So next, because TGF-beta is enriched in fibrotic liver, and we need to consider how TGF-beta modulate the cancer cell growth in fibrotic liver. Hepatic stress cells are the main producer of TGF-beta and induce the proliferation and activation of both MDSCs and Treg cells via secretion of amphiregulin. And as you see here, L6 in cirrhotic liver are responsible for TGF-beta dependent hepatic Treg cell induction and generate CXCL16 and control the accumulation of CXCL6 plus hepatic NKT cells. And furthermore, TGF-beta is known to directly modulate Treg cells and upregulating cell activity and promoting proliferation leading to CTL exhaustion. Thus, TGF-beta is contribute to the immunosuppressive environment against liver cancer development. Regarding the VEGF, also enriched in fibrotic liver, VEGF induced tumor angiogenesis, resulting in the malformed and malfunctional tumor vasculature. Tumor endothelial cells exhibit immunosuppressive characteristics such as PD-L1 expression, which enhance the exhaustion and apoptosis of T cells. I will not go into the detail about this aspect. Therefore, VEGF contribute to the development of immune suppressive tumor microenvironment in cirrhotic liver. So then I will mention about a classification of hepatocellular carcinoma. So in the 2019 fifth edition of the WHO classification of hepatocellular carcinoma, approximately 65% of all HCC are classified as not otherwise specified HCC, NOS HCC, without any distinguishing histopathological features. Whereas the remaining hepatocellular carcinomas are classified into the following 8 subtypes: steatohepatitic, clear cell, MTM, KRAS, chromophobe, fibrolamellar, neutrophil rich, and lymphocyte rich. So, for instance, here you see the steatohepatic hepatocellular carcinoma. This type is observed in steatohepatitis due to metabolic syndrome or alcoholic abuse, and characterized by macrovascular steatosis, balloon cells, intratumoral inflammation, and intratumoral pericellular fibrosis. In this type, as you see here, IL-6 JAK-STAT signaling is activated. However, lower frequency of catenin B1, TAT promoter, and the TP53 mutation was observed in this type. On the other hand, macrotrabecular massive hepatocellular carcinoma is commonly observed in SCV, derived hepatocellular carcinoma. This type is characterized by basophilic cytoplasm, macrotrabecular growth patterns, and enlarged hyperchromatic nuclei. So in this type, TP53 mutation and or FGF19 amplification, as well as overexpression of angiopoietin 2 and VEGF-A is common. So I don't explain other types of the liver cancer, but such kind of histological association with the gene mutation is very important to understand the HCC occurrence. So then, hepatocellular carcinoma is also can be divided into 2 major molecular classes. One is proliferation classes, the other is non-proliferation classes. So proliferation classes is characterized by more aggressive tumor with pure histological differentiation and the common association with hepatitis virus B-related HCC and high vascular invasion and elevated AFT levels. This type's genetic feature is characterized by increased chromosomal instability and frequent TP53 mutation and FGF9 and CCND1 amplification. A proliferation class is divided into 2 subclasses. One is proliferation progenitor cell subclasses, 30%, and proliferation Wnt TDF-beta subclass 20%. On the other hand, non-proliferation class is characterized by a less aggressive tumor with well to moderate differentiation, association with MASH, ASH, and HCV infection, a less frequent vascular invasion, and low AFP levels, and corresponds to S3 or I cluster 2 of the cancer genome atlas. This non-proliferation class is subdivided into Wnt beta-catenin CTNB1 subclass and interferon subclasses. So finally, I would like to introduce another classes of hepatocellular carcinoma. This is classified by immune class and subclass. So about 35% of hepatocellular carcinoma is classified as inflamed class, and they are divided into immune active, exhausted, and immune-like subclasses. The inflamed class is very common in patients with HCC who responded well to anti-PD-1 and PD-L1 antibodies. On the other hand, 65% of hepatocellular carcinoma is classified as non-inflamed class. And they are divided into immune intermediate subclass and immune exhaustive subclasses. So taken together, since the classification of hepatocellular carcinoma is closely related genomic abnormalities and molecular signaling, it provides important information in the development and selection of cancer treatment. So then I would like to talk about MASH-specific gene alterations in hepatocellular carcinoma. So globally, about 10% of all hepatocellular carcinoma caused are attributed to MASH-D, with the highest estimate report being studied from United States, United Kingdom, India, Germany, and the Middle East, and the lower estimates from China and Japan. Compared with patients with HCC related to viral hepatitis or alcoholic-associated liver disease, those with MASH-D HCC have a lower male to female ratio and 5 to 10 years older and more likely to have metabolic and cardiovascular comorbidities. As shown in the right panel, from 2002 to 2016, the prevalence of HCC in wait list candidates increased 11.5 in MASH. So the pathway to carcinogenesis in MASH might involve the release of inflammatory cytokines like interleukin-6, TNF-alpha, leptin, and resistin, and so on, along with the decreased amounts of adiponectin, leading to insulin resistance and increased exposure of hepatocyte to free fatty acids. Insulin resistance exhibits oxidation of fatty acids, leading to increased intracellular fatty acid, which cause oxidative stress to DNA and induce the genetic mutations. For example, the mutagen 4-hydroxy-2-nonenal, 4-HNE, is well known to contribute to hepatocarcinogenesis and has been associated with the mutation of codon 249 in a TP53 gene. So on the other hand, ethnically, it is known to be associated with the risk of ethnicity is also known to associate with the risk of hepatocellular carcinoma. So it was reported that genetic polymorphism in PNPLS3, TM6SF2, and GCKR and MBO87 have been associated with the increased risk of hepatocellular carcinoma, while HSD17B13 decrease the occurrence of hepatocellular carcinoma. However, recent report by Gerhard Christensen showed that genetic link score comprising 3 common variants in PNPLS3, TMF6F2, and HSD17B13 was associated with up to 12-fold higher risk of cirrhosis and up to 29-fold higher risk of hepatocellular carcinoma. Another report showed that the PNPLS3 RS738409G risk allele is observed in 40% of the European population and increased the risk of HCC development by 12-fold. Taken together, these findings indicate genetic predisposition plays an important role in the risk of developing hepatocellular carcinoma. So, as I mentioned, mutation in TAT promoter, catenin B1 and TP53 are the most frequent genetic alterations also in MASH, induced hepatocellular carcinoma. In addition, recently, Pignol and others reported that activin A receptor activin A receptor type 2A, ACVR2A, is the another frequent altered genes in MASH HCC, and incidence of ACVR2A mutations were higher in MASH HCC than in other etiologies. So furthermore, this report additionally shows the pattern of mutation signal must-seek MASH HCC, which was present almost exclusively in MASH HCC, compared to viral or alcohol-induced HCC. Interestingly, this signature was associated with small developing females. So thus, the molecular characterization of MASH-related HCC is not always the same with HCC due to viral infection or alcohol abuse. Further analysis of molecular characterization of MASH will require to develop diagnostic marker, prognostic marker, and therapeutic specific for MASH, MASLD, and MASH. So, ladies and gentlemen, here you see the summary of my talk. Thank you very much for your kind attention. Thank you.
Thank you, Professor Kawata. Excellent talk and nice overview of the genetic alternations. Is there a comment or question or suggestion from the audience? Or maybe Professor Kodama? Okay, please.
The ACVR2 gene is very interesting in the non-viral HCC. So do you study this gene in the Asian HCC patient?
I have no idea about that. Yeah.
This would be an important topic. Thank you.
Yeah, thank you very much.
Professor Kodama?
Yeah, before I'd like to ask you, Professor Kawada, for the question, I think I can answer the previous question, because I also published the papers for the non-viral HCC. We performed 113 patients of the sequence, and actually the NASH HCC patient have the higher percentage of ACVR2A mutation, so it's completely the same as the one that reported. So I think even in Asian patient also, it's also applicable. Okay, and very nice talk, and very comprehensive and very informative talks. I really enjoyed your talk. So first question is, you mentioned brief detail about NASH HCC profiling. So do you have any specific the difference between the NASH HCC and alcoholic HCC? Because you know that in Japan, alcoholic HCC incidence is rising. So.
Yeah, thank you very much for your good comment. Yes, actually, historically, NASH, MASH, and ASH is very similar. And also the gene mutation may be occurred by oxidative stress caused by intracellular oxidative stress. And maybe 4-HNEs are involved in the gene mutation, both in MASH or ASH-induced hepatocellular carcinoma. However, actually, in our laboratory, we have not analyzed yet about the gene mutation, and I read several papers, and they have not identified a specific gene mutation which is different from MASH and ASH-induced hepatocellular carcinoma. So maybe your comment, your question, maybe should be studied in the future. Thank you very much.
Okay. Okay, I think time is going out, so we are moving to the second speaker.
Thank you very much. Thank you.
The second presentation is Professor Nawakato. Professor Nawakato is very famous for paper study on improving virus hepatitis cancer. And the title is integrated the role of hepatitis B virus integration in hepatocarcinogenesis.
Good morning, and thank you for your kind introduction, Professor Masaki. So I'd like to start my presentation, and as you know, HBV is one of the major causes of HCC. About 300 million people are living with chronic hepatitis B infection, with 1.5 million new infections each year. And hepatitis B can cause a chronic infection and put people at high risk of death from cirrhosis and liver cancer. And hepatitis B resulted in an estimated 820,000 deaths, mostly from cirrhosis and HCC. This slide shows the HBV-induced hepatocarcinogenesis. As you can see, first, viral infection occurs, hepatocyte, HBV infection, and random integration occurs, and HBV DNA integrated into the hepatocyte. And the cis and the trans effect from integrated HBV and the genetic change, and when patient becomes cirrhotic, selective clonal expansion occurs, and also plus genetic change, especially oncogene increased and the tumor suppressor gene decrease, expression decrease, and finally malignancy development. In HBV DNA integration and hepatocarcinogenesis, as you know, HBV is known to integrate into the host genome during the course of infection and has been suggested to be associated with hepatocarcinogenesis. But the detailed role of HBV DNA integration in hepatocarcinogenesis is unknown. This slide is I used from Professor Ye and Professor P.J. Chen. And as you know, distribution of HBV DNA integration hotspot is telomerase and MLL4, KMT2B equal to KMT2B and cyclin 1, and also that promoter mutation exists also. And the distribution of HBV integration breakpoint in the TAT gene promoter and exon in HCCs, and also this slide shows the distribution of HBV integration breakpoint in MLL4 exons and introns. So we reanalyzed HBV integration sites in HCC. Basic data is published by Zhao et al. Nature Communications for 426 tumors. But this reanalysis used two new elements. T2T CHM13 is a newly published human whole genome, complete human genome sequence, compared to previous, including centromere and the telomere information. So we renewed one, and also virus break end is the latest HBV integration detection software. So as you can see, KM2B and TAT is a frequent spot of HBV integration, but we found new HBV integration clusters in the 11q 13.3 region. And unlike previous known gene integration site, such as TAT and KMT2B, 11q 13.3 spans multiple gene regions. And actually, we have found potential new loci associated with hepatocarcinogenesis. This 11q30.3 is, of course, enrichment of integration was observed. And CCND1, cyclin D1, FGF19, FGF4, FGF3 are located at this site where copy number amplification frequently occurs in tumors, not only HCC, every tumor. And also, this 11q13.3 is known for local copy number amplification in HCC. And this position and the red signal is amplification. And actually, 7% of HCC have local copy number amplification at 11q13.3. This is showing the distribution of oncogenesis, of oncogenes. And thus, HPV integration at this locus may be linked to cancer gene activation. Very recently, this is unfortunately not our work, but we are guessed like this because extrachromosomal circular DNA transmit DNA-based amplification occurred. And this is playing an important role in HPV HCC. HPV integration could take the form of extrachromosomal circular DNA. And this circular DNA amplifies inside the cells and resulting in elevated copy numbers and gene expressions. Until now, we can't understand how copy number increases. But actually, in case of hepatitis B virus integration, extrachromosomal or circular DNA, like plasmid, is increasing inside the cancer cells. This is published in Nature this year. And this is a second talk of my topics. HPV DNA integration, we often see, and actually integrated HPV DNA often contain X region and also lacking 3' end. So we get a new hypothesis because X region integrated, but the 3' end terminated. So transcribed messenger RNA is a fusion messenger RNA with HBX and human peptide. So fusion XBX consisting of HBX plus human peptide may be involved in hepatocarcinogenesis. So fusion messenger RNA identification was attempted from Hep3B cells established from B HCC. And actually, fusion messenger RNA exists with HPV sequence and human-derived sequences. And from this messenger RNA, maybe fusion XBX expressed, and this peptide is having HBX lacking C-terminal in N-terminal region. 61 amino acid derived from human sequences. So we have identified fusion messenger RNA with 3,725 base pair in length containing 1,877 base pair human derived sequence in this 3' end. And the fusion messenger RNA suggests the possibility of translation of fusion HBX with C-terminal deleted HBX followed by human-derived sequence. very interestingly, if we knock down this fusion messenger RNA, HPV cells upregulated genes in knockdown cells are included in ion stress response pathway. And also ATF family may be involved in the expression changes as regulators. So fusion HBx from HBV integrant affects hepatocarcinogenesis through deregulation of ER stress response. So HBx has a much function. Cells like regulation, DNA repair, regulation of apoptosis, activation of transcription factors, and also HBV replication, modulation of signaling pathways, and loss generation and calcium signaling. Thus, we thought that HBs could be a good target of direct acting antiviral. So now the program is currently used anti-HBV drugs cannot eliminate HBV. So there is a strong need to develop a new class of anti-HBV drug that can eliminate HBV. And the background is showing that HBx is a multifunctional protein and is essential for viral replication and carcinogenesis. So our hypothesis is that HBx binding compounds is possible to inhibit HBx's functions associated to viral replication and carcinogenesis. Thus we screened HBx binding compounds and analyzed their effect on HBx's function. This slide shows the method to screen HBx binding compound using FDA-approved drug on board chip and using purified GSD HBx, and analyzed using surface plasma resonance imaging, measures the angle and the strength of reflected light on the metal surface. So if bind graph become like this. So actually we use genotype B HBx and also genotype D HBx, and there are 8 compounds identified as genotype B HBx binding, and also 22 compounds were identified as genotype D HBx binding compound, and there are 6 same compounds between the two. So this HBx binding compound is tranilast, bromifene bromide, azithromycin, and so on. And the strongest was tranilast. At first, we evaluated anti-HBV activity, and before anti-HBV activity, we checked the toxicity of this compound, and the cell proliferation assay is showing that ALX-HC area is very toxic, so impossible to use for the drug. But the effect, this was showing the effect against HBV DNA. In Decavir like this, and the tranilast and the PDDB has anti-HBV DNA action. Interesting. And we checked the anti-HBV activity evaluation of tranilast. As you know, tranilast is anti-allergic compound, basically. And HBV DNA, not more than Decavir, of course, but tranilast decrease HBV DNA. And about HBs antigen, in Decavir, impossible to decrease HBs antigen. But tranilast can reduce HBs antigen, but cell viability no problem. So tranilast decrease both HBV DNA and S antigen level without affecting cell viability. However, tranilast had no anti-proliferation activity against H3B cells. So maybe tranilast bind to HBx, and our preliminary data suggested that decreases transcriptional activity of HBx, this compound. So decreases HB replication, but impossible to suppress hepatocalcinogenesis. So conclusion. HBV integration is involved in hepatocalcinogenesis. And 11q13.3 was identified as a new integration preferential site and contained oncogenes such as CCMD1, FGF19, FGF4, and FGF3. 11q13.3 showed increased copy number and increased expression of the included oncogenes. And HBV integrations could take the form of extrachromosomal circular DNA, resulting in elevated copy numbers and gene expression. HBV DNA integration into human genes often involves HBx, and fusion HBx made from the integrated HBx is involved in carcinogenesis. So from this data, we can explain. Everyone says HBV is associated with hepatocalcinogenesis through integration. But some says HBx is involved in the hepatocalcinogenesis. But this hypothesis may explain both of them. Integrated HBx and fusion protein has oncogenic protein. And interestingly, previous I showed the ER stress pathway signaling activate. But pure HBx has no such action. Only fusion HBx has this action. And finally, HBx binding compounds may have both antiviral and anti-cancer activities and may become a good target for future drug development. Thank you for your attention.
Professor Kato, thank you very much for very excellent presentation using your many published data. Thank you very much. There any question?
Professor Kato, thank you very much. It's an immature question, but
From the articles I have read, the integration number is not very high usually in HPV patients, the number of copies integrated. But in your expertise, what was the highest number of integrated sites per one person in your lifetime experience? And what was the lowest number? Thank you.
I don't know the lowest, but now we count because next generation sequence, as you know, we can't know the number, but we can know it by the number of breakpoint. And some patient more than 1,000 breakpoint. So many integration occurred. But this is basically randomly occurred. And only HCC clonal expansion, so we can detect the point where is the breakpoint. And actually, maybe more than 80 or 90% of patients with viral HCC has integration in liver cancer. And in my opinion, not number of the integrant is not so important. Most important thing is where to integrate. And depend on the site of integration, maybe oncogene expression increases or sometimes anti-cancer gene expression decreases. Thank you.
Please. Great talk. I was wondering, evolutionary, is there any relationship you can think of why HBV is integrating in 11q13 site?
Yeah. What's actually, we use sequence method, but usually what HBV DNA integration is difficult to check it, because as you know, patient has many free HBV DNA, so many HBV DNA sequence. So we have to do the special analysis about the integration site. Without it, we can't get the sequence information which HBX is really integrated and fusion messenger RNA appeared from there. So we'd like to do that for future plan. Thank you.
Professor Kodawa, please.
Thanks, Professor Kato, for the excellent presentation. This contains a lot of the important information. So I'm most interested in extra chromosomal circular DNA stuff.
Yeah, great.
And then you showed today that actually 11q region actually HBV DNA integrated and then involved in hepatocarcinogenesis, probably producing the FGF family proteins. So do you know whether these also FGF families are also expressed from the extra cellular circular DNA stuff? Do you know?
Yeah. Actually, we are checking it, but this year published, so it is very sorry for us, but maybe also 11q 13.3 was included in the extra chromosomal circular DNA. Very interesting thing.
Also, another thing, you didn't mention today, but I think that you know that HBV integration frequently occurs in fibronectin in non-tumor.
Yeah, right? I know.
Do you know the reason? I always wonder why it's fibronectin lesion so targeted.
Yes, of course, in our analysis, also integrated in fibronectin, but function of this integration is unfortunately still no explanation. So I don't know about that.
Thank you.
Thank you very much, Professor Kato.
Thank you.
And the third speaker, Professor Shinji Tanaka, he's a very famous surgeon in Japan. He's not make it today. He sent us a video instead, and next 20 minutes, we'll watch his presentation through the video. Thank you.
Thank you for giving me the opportunity to present our studies on integrated omics analysis for progression of hepatocellular carcinoma HCC. I have no COI to disclose. Today's agenda is shown here. Beyond surgical indication, advanced HCC with good liver function is indicated for cysteine therapy, and currently 8 regimens are approved in Japan. If these therapies are successful, it can be indicated for conversion surgery. More than 10 years ago, in the sorafenib era, this case was treated with sorafenib at Musashi Red Cross Hospital. After objective response was achieved, he was referred for surgical resection, resulting in good prognosis. With the advent of denpatinib with high tumor suppression effect, the number of conversion surgeries has increased, resulting in the improved prognosis. Recently, sequence therapy with denpatinib, sorafenib, and immunotherapy atezolizumab, our conversion surgery revealed that pathological CR was obtained. Conversion surgery after immunotherapy are also increasing now. Therapy for HCC has changed drastically from angiogenesis inhibitors to immune checkpoint inhibitors. And in this situation, as is in the whole, surgery can be used to play our best trump card after cysteine therapy, and also used as the option after adjuvant therapy, as demonstrated by Embrave 050 clinical trial. There are various therapeutic strategies, but we can't select now what kind of combination in which order for which type of HCC. So, biomarkers are required for this selection. According to previous studies, in general, one of the important biomarkers associated with response to immunotherapy is tumor mutation. In HCC, based on the third promoter mutations, TP53 and CTNP1 beta-catenin mutations are mutually exclusive, and ALT2A, ALT2 mutations are relatively common with impaired chromatin remodeling. We reveal that ALT2 knockout in HCC cells by genome editing could lead to disruption of DNA repair, resulting in high tumor mutation burden in HCC patients. But the tumor mutation burden is not usually recognized as the biomarker for immunotherapy in HCC patients. Another candidate of the biomarker is inflammation, that is hot tumor or cold tumor, analyzed by gene signatures. For example, Hoshida S3 signature contains inflammation subclass, and Boyert G4 subtype is carried by inflammatory signals. So we utilize our own surgical samples of HCC to analyze transplant data and genome editing, genome and epigenome data for omics studies. We revealed that HCC can be classified into 3 molecular subtypes: MS1 carrying P50 mutation, chromatin remote instability and MYC pathway, MS2 carrying beta-catenin mutations and DNA methylation phenotype, and MS3 carries by the metabolic syndrome. Tumor tissues, as you know, consist of not only cancer cells, but also various immune cells as tumor immune microenvironment. In this regard, from bulk tissue data, the cyber sort method is used for deconvolution of immune cell subsets. The cyber study revealed that significant immune suppression was recognized in the MS2 subtype that is cold tumor with mutant beta-catenin mutation. Especially, CD8 set T cells are extremely decreased by beta-catenin mutation. So we used multiplex genomic editing technology in inducing beta-catenin exon 3 skipping for inflamed mutation to construct MS2 cold subtype HCC. The syngeneic genomic models showed cold tumor growth without cytotoxic T cells, and we identified that CCL20 was down-regulated in all our model, and as well as in human samples of beta-catenin mutated HCC, and we revealed that CCL20 upregulation can recover the activity of dendritic cells and cytotoxic T cells. CCL20 is known as one of the powerful activators of dendritic cells, and the cytokines stimulation might be a promising approach for treatment of the cold subtype HCC. Indeed, according to clinical studies, beta-catenin mutated cold HCC are insensitive to immune checkpoint inhibitors. So, recent studies reveal that RNA medicine is useful to deliver the cytokine cocktail to activate the local anti-tumor immunity, as well as abscopal effect on metastatic tumors. As you know, the efficiency of RNA medicine is proven by many clinical trials for COVID-19 vaccine. RNA medicine is a promising approach to treat cancer patients. Currently, we are collaborating with the specialists of RNA medicine to develop the novel methods for induction of a potent abscopal effect and for clinical application to treat the cold tumor HCC. In addition to MS2 type, we focused also on MS1 carrying TP mutation and MYC activation with highly metastatic potential. To construct MS1 subtype of HCC, we induced CRISPR-Cas9 knockout, p50 knockout, and MYC overexpression, resulting in chromosomal instability and immune resistance. It should be noted that this model demonstrates highly metastatic potentials and macrotrabecular massive MTM structures characterized as a clinically metastatic phenotype of aggressive HCC. And we found a biomarker X1 and also developed X1 targeted therapy and combination of immunotherapy. In our clinical subtype of HCC, we identified MS1 specific therapy and MS2 specific RNA medicine. MS3 subtype contains a subgroup enriched with regulatory Tregs. Indeed, according to Dr. Nakagawa and our collaboration studies, Treg high subgroup of HCC showed poor prognosis. Recent study revealed that Treg can be enhanced by lactate in highly glycated tumors with immune microenvironment, immune resistance, but lactate transporter MCT1 can recover the sensitivity to immunotherapy. As immunosuppressive microenvironments, we must focus on the glycolase and lactate, And we identified X3 mutated HCC closely associated with Treg enrichment and prognosis. And knockout of X3 gene in HCC upregulates lactate and indicate Treg accumulation. In vivo resistance to immunotherapy was abolished by MCT inhibitor. As shown, a model of Treg high HCC might be susceptible to inhibition of lactate transporter MCT. Indeed, one of the specific MCT inhibitors has been recently used in phase 1 clinical trial, so some combined immunotherapy might be a promising approach for the specific subtype of cancers. Based on recent advances in system therapy in HCC, we established the molecular and immunological subtyping, and we are now developing the subtype-specific therapies. This field is selected as a highly selected cited paper placed in the top 1%. More recently, we are trying single-cell analysis and another new target gene X for novel therapy of advanced HCC. In the cancer immunity cycle, tumor immune microenvironment might play an important role in subcycle. And we reproduced all of the subtype model mimic to metastatic MS1, cold MS2, and immune-resistant MS3 to develop specific inhibitor and RNA medicine for potentially clinical application. Genome and RNA medicine might overcome the aggressive cancers including HCC and pancreatic cancer. In summary, to construct the precise precision therapy, it's required to clarify the tumor immune microenvironment time in HCC. Based on molecular and immunological subtyping of HCC, the subtype-specific immunotherapy can be developed with immunocompetent mimicry models. Genome and RNA medicine, as well as single-cell time analysis, should bring about a paradigm shift in treatment strategy for HCC patients. My collaborators with study networks are shown here. Thank you for your attention.
Okay, last paper will be handled by Professor Cao, please.
I'm honored to introduce Professor P.J. Chen, who is a professor of Hepatitis Research Center at National Taiwan University Hospital, Taiwan. Professor Chen is a very famous research scholar in infectious diseases, especially in viral hepatitis, HCC, both in basic and clinical research. Professor Chen, please.
Okay, okay, thank you, Professor Zhou. So I'm the last talk of this session. So I will not try to confuse you with complicated data. We are trying to, I think I will give you some of the overview and especially the challenges and the future aspect. Can you turn off the light? Yes, okay, thank you. So in this session, I will discuss with you some important idea about the current strategy and the future prospect. Here is my disclosure. As you know, for the HCC, currently, especially the genomic, the knowledge gaps and the unmet needs is very simple. Scientifically, we know there are many mutations. But however, what are the most important driver mutation? This is still the question, not answer. Medically, what are the clinically useful or actionable genomic mutation in terms of the classification, diagnosis, or treatment? You saw many kinds of the classification, but none has been implemented in the clinical setting. So in this review, the summary, the most common somatic mutation, such as the third promoter mutation, p53, beta-catenin, and etc. But however, the detailed analysis for the stage of this common mutation taking place is usually after the stage of the early hepatocellular carcinoma. So this is mean this common mutation are the late mutation. So they amplify the carcinogenesis. In fact, in the early stage, dysplastic nodule or the cirrhosis, there are very few mutation has been recovered. So, and the mutation at this stage are very important. It is similar to the APC gene in the colon polyp. They are the fundamental mutation, but we have very limited information regarding to the somatic mutation in the precancer stage of the HCC, especially the cirrhosis. A few years ago, a group in UK studied the genetic mutation in the cirrhosis. They conducted micro dissection for this cirrhosis nodule and analyzed the whole genome sequence. And it's very surprising, they found that there's no significant mutation in the cirrhotic nodule. The only one is I just mentioned to discussed before. One is the albumin, but albumin, you know, this is not relevant. The only significant one is the ACVR2A, but this is, again, it's a very small proportion. So the driver mutation in the precancer cirrhosis, actually, we know very little. And so what happened, what kind of the mutation or the genetic alteration happening in the precancer cirrhosis, we really don't know. Is this are there epigenetic alteration we don't know, or we have the found copy number variation, or growth but the recurrent translocation. This one, like mentioned by the Dr. Kato. Or the more important is there is a common cancer-promoting liver microenvironment. This is very interesting. As you know, the chronic hepatitis is the most important predisposing factor for promoting HCC. Therefore, if you treat the chronic hepatitis B or hepatitis C well, we can delay the carcinogenesis significantly. So we don't know which one is important, but this one is clinically very significant. But regarding the early or the driver mutation in the precancer stage, like the chronic hepatitis or the cirrhosis, the best example is the HPV-related HCC, especially the HPV integration, as Dr. Kato just mentioned. The HPV integration happening in the is a very unique genomic mutation for HPV-related HCC. This HPV integration have been found in about 90% of the HPV of the chromosome of the HPV-related HCC. So this is very common, more common than third mutation, promoter mutation for HPV-related HCC. But this HPV integration is not a mandatory step for the HPV life cycle. This is different from the retrovirus. So HPV infection, after the infection, the DNA release and then converted to the CCC DNA, and this CCC DNA is a template for the transcription and the translation and eventually the virus production. So this episomal pathway is the HPV life cycle. But incidentally, the HBV DNA can be converted to the double-strand DNA, and this double-strand DNA can integrate into the HBV, into the chromosome or the infected hepatocyte by DNA repair mechanism. But so this is not a mandatory step in the HBV cycle. Therefore, the success rate, the integration rate of the HBV DNA into the chromosome or the infected hepatocyte is very low. Here is a Thomas study. Immediately after the infection, in the hepatitis 2 cell or the HU7 NTCP cell line, you can appreciate the integration can be noted after 1 week after the infection. But the rate is very low. It's only 0.01% to 0.1%. But however, this integration event apparently taking place much earlier than the cirrhosis or in the HCC. So if we review the progression from the acute HBV infection to the hepatitis, cirrhosis, and HCC, HBV integration taking place at this stage, and they only happening in about 0.1% or even lower, or the hepatocyte. But however, in the established clinical HCC, HBV-related one, they are 90% containing HBV integration. Therefore, the hepatocyte with HBV integration apparently have the advantage to grow into the clonal expansion, then eventually to the HCC. So the proportion increasing from the 0.1% to the 90%. So the HBV integration is very likely a possible driver mutation for the HBV-related HCC. So this is I'm just propose the study of the HBV integration in the cirrhosis stage or even in the earlier stage may be very important to understand what are the likely driving force to become the HCC. So this part, the study are very limited, and I think we should pay more attention and using the HBV integration as a probe, so we can understand what happened to the chromosome or the hepatocyte in the precancer stage. The next, I'm discussing about the genomic mutation profile for the HCC classification. There are many classifications. I cited this one from the Dr. Jessica Zuckerman. She published many important genomic mutations of the HCC paper, and then proposed several classification. And you know, there's a Hoshida classification, and Dr. Tanaka, and there are many kind of classification. But unfortunately, there's no one become clinically useful. So we really need to know what we can do better. What the classification can make the clinical sense in the future. So to this regard, I would like to discuss with you the classification for the HBV-related HCC basing upon the HBV integration. And there's a third promoter mutation. We have studied the HBV integration among about 153 cases, HCC, and each bar is one case. And you can appreciate about 88% of the patient, they are HBV DNA integration in their chromosome. And there are two hot spots. One hot spot is HBV integration into the HBV TERT promoter. This accounting about 1/4 of the patient. There's another hot spot is integration into the exon 3 to 6 of the MLL4 gene. This proportion is about 10 to 15%. But interestingly, this has been noted before. For the HCC, HBV-related HCC, the group with integration into the TERT promoter or into the MLL4 exon 3 integration. These two groups, they are mutually exclusive. They don't they don't overlap. And recently, we discovered there's a certain mutation. This is the TERT promoter point mutation. This is 124 point mutation. Again, we noted very interesting. These 3 mutations are happening in the different subgroup of the HBV-related HCC. So far, we did not find a case with the overlapping genetic mutation of this, between the 3. So in our mind, for the HBV-related HCC, we can rely on the genetic mutation dividing into the group 1 with the HBV integration into the TERT promoter, group 2 into the MLL4 exon, group 3 with the TERT promoter point mutation. Group 4 is devoid of the 3 mutations. So we use the 3 somatic mutation, can divide HBV-related HCC into 4 independent groups. And there's some interesting biology. For example, we know the group 3, these patients usually are older. And the group 4, they are less male dominant. As you know, the HBV-related HCC, they are male dominant. But in the group 4, the gender difference is not much. Furthermore, we also noted this is for the post-operative HCC. For the group 4, usually the survival, or the recurrence-free survival, or overall survival, they are much better than other 3. So we are exploring whether the HBV integration, the 2 integration and 1 somatic mutation can be a new classification for the HBV-related HCC in the future. And in addition, because this mutation, they are mutually exclusive. So we are quite interested to see whether the HBV integration into the TERT promoter or into the MLL4, whether they are independent carcinogenic pathway, and whether we can recreate this pathway and their categorical HCC in the mice model. So we are currently working on this, and we have some interesting data. Yes, we can develop the HCC model depending the TERT HBV integration into the TERT or into the MLL4. They're showing different cancer property. And finally, I also want to discuss with you the HBV integration can be a very useful personalized circulating tumor DNA for the HBV-related HCC. This is because the HBV integration site for individual HCC are unique. So every HBV-related HCC, if they have HBV integration, their integration site, especially their specific junction sequence, these are unique to individual HCC. And this junction sequence, as you fully aware, this is a somatic mutation. And if this fragment can be released into the blood, into the circulation, they become signature signature circulating tumor DNA for this HBV-related HCC. So we can take this advantage and start checking the junction sequence, this consisting of the viral sequence and the host sequence. So we call this the viral host chimera DNA. We can detect this chimera DNA in the blood or HBV-related HCC patient. In fact, we can successfully develop the digital PCR using the junction sequence for individual HBV-related HCC, then measure the amount or how much of the junctions DNA sequence has been released and present in the blood of the patient. And we found that the level of the circulating viral host chimera DNA are in proportional to the tumor size. And in some cases, it's more sensitive than the alpha-fetoprotein level, especially for the small tumor. In the small tumor, the positive rate for the chimera DNA is 90%, but for the alpha-fetoprotein is only about 40%. So the sensitivity is much better. So finally, this is the progression from chronic hepatitis B to cirrhosis to the HCC. And I just mentioned to you the circulating chimera DNA from the HCC can be detected in the blood of the HCC patient. But however, if we can improve our sensitivity to detect the viral host chimera DNA much better, then we probably can detect the clonal expansion even before the HCC. So we can detect the chimera DNA in the blood or in the patient of the cirrhosis or even in the chronic hepatitis. But this will need much more effort in the future. So the circulating viral host chimera DNA in the HBV-related HCC, they can be used as a marker to detect any residual cancer cell after the local curative treatment. Or we can follow the response to the systemic therapy. So this will be an interesting biomarker to be explored. And finally, this is a very old stat, about maybe 20 years ago, by our mentor, Professor D.S. Chen. Long time ago, he already indicated that HBV integration in the future may be a very good, even a good and more useful marker than the HBs antigen for the detection. So I think so we are happy we can fulfill some of his prospect. So with this, I would like to stop here and summarize. I just want to emphasize HBV-related HCC is a good example for study the driver mutation and the condition. So, this can be applied for the HCC classification, and even to develop the animal model, so to help developing HCC-specific target in the future. Finally, the circulating tumor DNA in the form of the viral host chimera DNA may be very useful in the future setting. So with this, I would like to thank all my mentor and also our collaboration team on this interesting topic. Thank you for your attention.
Thank you very much, Professor Chen, to present us the gaps and unmet needs and challenges and the futures in the unraveling the progression of genomic abnormalities of HCC. Very interesting and perspective, very wonderful presentation. now is open to comments.
Excellent talk.
To the
And
audience.
Enomoto from Osaka Metro. And you mentioned chimeric DNA can be detected in almost all HBV HCC patients. Do you mean it can be detected in very early HCC or can be used for early detection of HCC?
Yeah. At this stage, we can detect the chimera DNA not only in the HCC bigger than 5 centimeter. If you don't know the chimera DNA, the sequence of the detection, you have two ways. One is directed from the plasma without the knowledge of the chimera DNA or the junction sequence. The other is if you know the junction sequence from the HCC, you can develop specific digital PCR for this junction. Yeah. Okay, so for the for the plasma direct capture, we can only detect about the 5 centimeter HCC. But if you know already know the HCC and the junction sequence from the HCC, we can detect about 2 centimeter, less than about 2 to 1.5 centimeter. Okay.
Thank you.
You're right.
Maybe I can answer your question because in our clinic, usually the ctDNA is maybe really work on the early diagnosis for HCC recurrence. Even because I'm a liver surgeon, after liver resection or even after transplantation, we monitor the ctDNA volume in the blood. If the ctDNA volume is increased, maybe after 3 or 6 months later, maybe imaging CT or MRI can find the recurrence. So now in this stage, if the ctDNA volume increase, we will do enhance the anti-cancer treatment. Maybe, but this is a preliminary result.
Yes, we also conduct a prospective study for the chimera DNA in the post-operative HCC. We will present the data in this afternoon. You're welcome to see. Okay.
Thank you.
We need to
Okay. You summarize, and maybe Professor.
You can summarize briefly.
Okay. Dr. Kodama summarize or
briefly.
Host the discussion.
Yes.
Okay.
Just summarize the session. Youngsters.
So it's summary, no question, right? So
okay, question. If you have question first, only one question. For time limitation.
Yeah, thanks for the excellent presentation. Actually, so you showed that in the first talk that you showed that actually the HBV integration pattern can be used for the molecular classification, and also you can show that the circulating HBV DNA can be used for the detection of the HCC. So in the future setting, if the sensitivity becomes improved, so do you think that this ctDNA stuff can also actually see the molecular profiling? Is that the goal?
You point out, I think the circulated DNA, because we just look at the chimera DNA, so we can only follow or tracking whether the clone is expansion or is it disappearing. But we cannot detect the other somatic mutation. Because this other somatic mutation is much more, the sensitivity to detect the somatic mutation is very low in compared to the viral host chimera DNA. So only for the clonal expansion cannot give the profile or all mutation. Yeah.
Thank you. And I need to summarize, right?
Yeah, so, just brief.
Yeah, so as a discussant, I was very interested and enjoyed this talk. The Professor Kawada actually showed us the global overview of the HCC genomic abnormality and also focus on the MASH HCC. And and Professor Kato mainly focusing on the HBV, the integration, the meaning of the hepatocarcinogenesis, and following the Professor Chen also showing us the HBV integration pattern, also future direction to the detection of the HCC using the HBV integration stuff. So, and also Professor Chen show us what is so far the unmet needs and what we have to further study for the this genomic abnormality in HCC. So I think this session we learned a lot, and we also learned a lot about what we have to do in the future perspective. So I think this is a very fruitful session, and I really appreciate all the patient participant active discussions. Thank you.
Excellent summarize. Thank you very much for participating in this session. This session will be closed. Thank you.
Thank you very much, the lecturers and moderators. Now we will have 1 hour and 20 minutes break.